I'd like to particularly acknowledge the inclusion of the needs of children and young people with developmental disabilities in this conversation, because you could argue that they are perhaps even more vulnerable to some of the early influences through early childhood and pregnancy. I'd also like to thank Ray Davison for his reminder to us that this is part of a conversation, and to say that I'm really pleased to see some young people in the audience who are being included in this conversation today. So this picture shows pranayama, which represents a direction that artist and neuroscientist Greg Dunn has been taking that explores alternative ways of interpreting the mind and brain. It is directly inspired by his practice of meditation and experiences inside his sensory deprivation tank. How interesting. Greg has completed his doctorate in neuroscience from the University of Pennsylvania and now creates beautiful artwork of the nervous system. For me, this not only beautifully and elegantly depicts the connection between the mind and body that are often neglected in our clinical practice in medicine and how our systems, health systems and other systems connect together, but also that the neurological system is dramatically influenced by the external world. This is especially so for the developing child. Some of the early part of my talk will reflect some of the themes that have already been very eloquently explored by the previous two speakers, but I've included them nonetheless because they really are the foundations in our understanding of child development, and they are just as relevant for children who will have a developmental disability as other children. So, Liz has already very clearly talked about the developing brain, and that a dramatic part of the development of the brain occurs through pregnancy. So as you can see in the picture on the left side, the earliest part of the brain is but a bubble at the end of the neural tube. But by the time the child is born, the brain already has about all the neurons it will ever have. It doubles in size in the first year, and as Liz mentioned, by the age of 3, it will have reached 80% of its brain volume. We've already heard about the importance of connections between the brain cells, and that during the first few years of life, 700 to 1,000 new synapses form every second. That's a phenomenal rate of change. After the period of rapid proliferation, these connections are reduced through a normal process called pruning, which enables remaining brain circuits to become stronger and more efficient. Those remaining are the ones that we use throughout life. This picture is a simplified diagram of the neuron. So as you can see in the picture on the left, the cell body has a number of arms that form branch-like structures that connect to other cells. Those connections are vitally important, and progressively we're understanding some of the complexities of the synapse, the space between the brain cells, and how those particular chemicals and proteins form such an important part in many diseases that develop and conditions that affect the brain. It's already been mentioned that skills beget skills, and that both positive experiences and negative influences are important for how the brain develops. Exposure to stressful life circumstances or environmental toxins leave a chemical signature on the brain and the genes. So we know that the skills beget skills as brain are built from the bottom up, with increasingly complex circuits building on simpler circuits, and increasingly complex and adaptive skills emerging over time. Because the circuits mature sequentially, different kinds of experiences are critical at different ages, and both Liz and Michael have already talked about that. Adversity impairs development. We've heard already that there's a cumulative effect of adversity that affects development through the early years of childhood, and that it's become increasingly clear that toxic stress can be seen in the brain architecture. And as you can see on the right, the neurons damaged by stress have fewer brain connections, those vital connections that are typically made through the early childhood years. The rest of my talk is going to touch on some key issues that are emerging as important for people with intellectual disability, and what the science is starting to show us. But firstly, I'd like to remind us all that it is important to consistently acknowledge that disability depends not only on the child's health conditions or impairments, but also crucially on the extent to which environmental factors support the child's full participation and inclusion in society. Children and young people with disability deserve the same rights to participate in their communities. However, for the remainder of this talk, I'll focus on some of the biological factors that contribute to intellectual disability. Intellectual disability can be caused by environmental factors such as alcohol abuse during pregnancy, infections, birth complications, and extreme malnutrition and traumatic brain injury. The pictures on the left show a normal child's brain, and the brain on the right is a brain that's been affected by fetal alcohol syndrome. You can see the devastating effect that alcohol has on the brain development. However, genetics is known to have an important role for many children and may well be an underlying risk factor for children exposed to other stressors. The brain is an incredibly complex organ consisting of a myriad of interconnected cell types. During development and day-to-day functioning throughout life, numerous proteins need to be functionally active in the right amount, at the right place, at the correct time. It is therefore not unexpected that a mutation, deletion, or rearrangement affecting one of the genes encoding these proteins can have a severe consequence for brain development or cognitive functioning. So the causes of intellectual disability may differ with different levels of intellectual disability. Reichenberg and colleagues published their research in 2016. They evaluated the hypothesis originally proposed almost 1 century ago that most intellectual disability is caused by genetic and environmental factors responsible for the normal distribution of intellectual IQ, or intelligence, but that severe intellectual disability is not. They studied more than 1 million sibling pairs and 9,000 twin pairs assessed for IQ and the presence of intellectual disability. They evaluated whether genetic and environmental influences at the extremes of the distribution are different from those operating at the normal range. They showed that factors influencing mild intellectual disability, the lowest 3% of IQ distribution, were similar to those influencing IQ in the normal range. In contrast, the factors influencing intellectual disability, the lowest 0.5% of the IQ distribution, differ from those influencing mild ID or IQ scores in the normal range. Taken together, their results suggest that most severe intellectual disability is a distinct condition, qualitatively different from the preponderance of intellectual disability, which in turn represents the lower extreme of the normal distribution of intelligence. In general, research suggests that mild ID is likely to be caused by many genes exerting small effects. One possible mechanism for non-inherited severe ID could be de novo point mutations, those mutations that occur for the first time in that child. They might occur in the germline, those mutations might occur in the germline during early pregnancy. Molecular genetic studies reported associations between those rare de novo point mutations and what proportion of severe ID they account for. Imprinting may also be involved. That's a form of gene regulation in which gene expression depends on whether the gene was inherited from either the male or female parent. These graphs show the importance and relevance for policy for our understanding and support of children and their families. The graphs on the left with the blue line show that where there's a child with a mild intellectual disability, it's very likely that their siblings may have an intermediate level of intellectual abilities. That is, they may have intellectual abilities lower than that typical for the population. That's very important if we're taking a child and family health approach. However, for children with a severe intellectual disability, the profile of intellectual abilities of their siblings map onto that of the general population. If we're looking at the genetic causes of intellectual disability, there's been a tremendous growth in our understanding of the genes that underpin intellectual disability. With the introduction of next generation sequencing technologies, I'll have to declare I'm not a geneticist, so I'm not the person to explain what these all are. New ID genes are now being identified in rapid succession. This combination of novel technology and increased biological understanding is rapidly increasing the diagnostic yield of genetic tests in intellectual disability and improving the usefulness of genetic tests for patients and families. So we can now see from this graph that there are over 700 identified genes associated with intellectual disability alone or with other features. Sometimes we see other physical features associated with intellectual disability, sometimes they're not present. And even on the X chromosome alone, girls have 2, boys have 1. Mutations in over 100 genes have now been found to be associated with intellectual disability. And you can see in the 1990s there were very few causes of intellectual disability for which the genetic cause was identified. So if we're thinking about autism spectrum disorder is a lifelong developmental disability defined by diagnostic criteria that includes difficulties in social communication and social interaction, and restricted repetitive patterns of behaviour, interests or activities. Initial signs and symptoms are typically apparent in the early developmental period. However, social deficits and behaviour patterns might not be recognised as symptoms of ASD until a child is unable to meet social, educational, or other important life stage demands. This may be particularly important in the justice system, where the difficulties of autism have not been recognised but have influenced their response to social demands and peer pressure. Also, functional limitations vary among people with autism and they may develop over time. One feature that's been observed in children with autism is that there seems to be greater brain growth through infancy than with typically developing children, as you can see from these pictures. This doesn't mean, however, that taking a head circumference of a young child is necessarily diagnostic or predictive of autism. But as Liz and Michael have mentioned, taking a population approach can help us better understand the development of the child and the brain, and particularly for children with disabilities. There are no medications that can cure autism or treat the core symptoms. However, there are medications that can help some children with autism with some of their difficulties. Researchers are continuing to explore different ways of helping children with autism. Fragile X syndrome is an example of... Fragile X syndrome is an example of a health condition that has implications not just for the child but also for other members of the family. Fragile X syndrome affects 1 in 4,000 males and 1 in 6,000 females and is the most common neurogenetic neurodevelopmental disorder, as well as the largest single genetic contributor to autism. The most common cause of Fragile X syndrome is the expansion of the CGG, trinucleotide repeats in the FMR1 gene to over 200 repeats. So I know that's a lot of jargon, but you can see through the graph on the right that once you get above 200 repeats, the Fragile X syndrome is created. Radi Hageman and colleagues in 2009 reviewed Fragile X syndrome and highlighted the importance of this condition not just for the child but also the family. In the carrier mother, who usually has a normal IQ, there is an increased risk for early menopause, depression and anxiety, and late onset neurological problems, including problems with functioning thyroid, fibromyalgia, problems with the nerves, and the Fragile X associated tremor ataxia syndrome. In the grandparent, who is the carrier, this is the grandfather, there is a high risk of the FAXTAS, the Fragile X tremor and ataxia syndrome, and dementia. FMR1, the protein that's coded by the gene, is involved in the control of protein synthesis, and recent studies have found that FMRP shuttles from the nucleus in the cell to the cytoplasm. The use of targeted treatments is an exciting area of research, particularly in Fragile X syndrome, and it holds the promise of reversing the epilepsy, behaviour problems and cognitive deficits in Fragile X, because these changes have been demonstrated in animal models with the use of the mGluR5 antagonist. However, as yet, despite the success in mouse models, no specific treatment has proven effective in people with Fragile X syndrome. Even though our knowledge of genetics has increased dramatically over the last 20 or 30 years, the distinctions between what we have drawn between conditions such as autism and mental health are still being challenged. In addition to ASD, autism and intellectual disability, copy number changes in the genes have been identified that are risk factors for schizophrenia, epilepsy and attention deficit hyperactivity disorder. That is, the same genes may code for risks for a variety of developmental and mental health conditions. There is substantial overlap in the number among the copy number variations in the genes that have been identified in each of these disorders and in cases of intellectual disability and autism. For example, small deletions on the chromosome number 15 have been associated with intellectual disability, autism and schizophrenia, and occur with increased frequency in people with generalised epilepsy. So other people such as Owen and colleagues have discussed the implications of the neurodevelopmental hypothesis of schizophrenia and that it can now really start to be fully appreciated. Recent studies indicate genetic overlap between schizophrenia and syndromes in which the psychopathology or the features are manifest in childhood and that are often grouped together as neurodevelopmental disorders such as autism, intellectual disability and ADHD. So the traditional distinction between adult onset, schizophrenia and childhood disorders is continuing to be challenged. So together with the evidence of frequent comorbidity, that is, they often overlap, these conditions may be seen in the same child, would suggest that a review of the functional psychoses as a member of a group of related and overlapping syndromes is important from both the clinical practice as well as the policy and configuration of services. So importantly, we are finding more answers for parents. This graph presents a picture of the diagnostic yield for moderate and severe intellectual disability, excluding Down syndrome, which represents 6 to 8% of all intellectual disability over time. The solid line indicates the mean of published studies, and the shaded background indicates the lower and upper boundaries of reported diagnostic yields. So you can see in the 1970s, the conventional testing or karyotyping, which was a routine test at that time, provided information for parents about the cause of their child's disability in only 3 to 6.5% of cases. There's the development of technology that it's included fluorescence in situ hybridization and genomic microarrays, and more recently whole exome sequencing has led to a dramatic increase in the number of genes we've been able to identify and provide answers for parents about what has caused their child's disability. So we are now up to our diagnostic yield of 55 to 70% for people with moderate to severe intellectual disability. Generally, we have a higher yield of finding the genetic causes for children with a moderate to severe intellectual disability than with a mild intellectual disability. And there's also differences in the diagnostic yield between boys and girls. In general, parents want to know what's caused their child's intellectual or developmental disability, what they can do to help their child, and will it recur if they have other children. With progress in finding the genetic causes of intellectual disability, we're better placed to provide this information for them. For many years, the prevailing view has been that intellectual disability cannot be cured because the effective biological processes, including neurogenesis, are difficult to treat and the cellular damage cannot be undone or reversed. Much of the treatment has therefore focused on environmental optimisation, including individualised education plans, as well as minimising the comorbidity, those things that we know might also go along with intellectual disability. such as visual problems, sleep problems, or pain that may make things worse and harder for children with disabilities to learn. For specific syndromes associated with intellectual disability, some therapeutic strategies are known. For instance, in a few metabolic disorders, enzyme replacement therapy is used, which can dramatically change prognosis and is sometimes accompanied by unexpected intellectual sparing. That is, you can actually prevent the intellectual disability. Recent studies involving specific types of intellectual disability, for instance those arising from specific genes, suggest that intellectual disability might be amenable to therapeutic intervention. This has dramatically transformed my understanding of what we can do even if there's a genetic underlying cause of the disability. I think 10 or 15 years ago we would have never considered treating conditions for which there was a genetic basis. Conditions such as Fragile X syndrome, Rett syndrome, MECP2, duplication syndrome, Dravet syndrome, and the list goes on, are being investigated in mouse models to see if new treatments can be developed and be useful for children. So the key points I'd like to share today are that our growing knowledge of brain science for children with disability tells us that diversity matters for them just as it does for children in the general population. We know that genes play very important roles in the emergence of intellectual disability and probably for learning and development intelligence in the general population as well. And that our new knowledge has confirmed that genes and environment interact, and this is particularly important for children with disability. So we're finding out more about the causes of intellectual disability, and this is particularly important for a range of reasons. We know that there's an increased likelihood of other people in the family having similar conditions, and this is particularly so if the first child has a mild intellectual disability. It's demonstrating to us that there's a clear relationship between different conditions such as mental health problems, autism, and intellectual disability, and challenging our separate systems that treat these conditions as different without recognising common causes and common underlying factors and common issues that make life more challenging for people with these different conditions. We can better understand what the health risks might be for the child and what the future might hold. And excitedly, there may be new treatments for children with an intellectual disability in the future. Importantly though, now we've got growing capacity to respond to parents' important questions for their child, and I think we have an obligation to continue to explore what we can to meet the needs of the parents and the child. And I know many of you will be aware of Bruce Perry and his profound work in the importance of early childhood years. So I'd just like to reiterate the remarkably dynamic process of brain development in early life offers a unique and fleeting opportunity to influence the health and welfare of the individual. And these are some important resources that I think have already been referenced through the other two talks and really are worth looking at more extensively. Thank you.